And thank you for those of us joining. And also thank you to the folks at RareSite who invited me to give a talk. So thanks. I'm looking forward to this and hope you all find it interesting. This is the title of my talk. And basically, I'm going to talk about our work in circulating tumor cells, how we got into this, and sort of where things stand at the moment. These are the objectives of the talk. Well, I'm going to start off talking about minimal residual disease and cancer in general, and talk about some technologies that can be used to try to find small numbers of cells. And then hopefully convince you that this technology will have a role in clinical management of cancer patients going forward. And I have received research funding from RareSite, which helped us actually do the validation study that I will be talking about. So, we talk about personalized medicine now for taking care of cancer patients. At one time, basically, we had tools that included chemotherapy, surgery, radiation, and that's about it. In the 1970s, people began using stem cell transplantation for hematological malignancies, and that's pretty much routine now. But really the things that have sort of moved cancer therapy forward and are sort of a little more revolutionary, if you want to put it that way, are targeted therapies. And so as we've learned more about the biology of cancer cells, we can do things like design drugs that interfere with the pathways that give tumor cells a growth advantage. We can use the immune system to try to eliminate cancer cells. And basically all this is done, a lot of this information that we've gotten has come from studying the genetics of cancer cells, and that in turn has been due to a lot of next generation sequencing and really finding out about, being able to look at large numbers of mutations at the same time. So one of the things that, when you're thinking about cancer is what does it mean to actually cure cancer? So what these four graphs here are showing are, if you have breast cancer, it basically the lines on each graph talk about some of the genetics of the cancer cells, and then each individual panel represents the stage of cancer. So if you have low stage disease, stage 1, as you can see, the outcomes are actually very good. We're very good at taking care of patients with stage 1 disease. But by the time you get to stage 4, which is metastatic disease, you can see survival is much more dismal, particularly in the case of breast cancer, the so-called triple negative patients who are both who are negative for hormone receptors and are negative for HER2 and so on. So we still have a way to go, particularly in trying to figure out what we can do for patients who have more advanced disease. And so one of the concepts here to think about is minimal residual disease. That is, you can treat a patient for cancer, and maybe you can see sort of at the level of X-rays or mainly imaging studies that a patient's cancer has gone away as far as you can tell. But it still doesn't mean that there isn't small amounts of cancer that are just evading detection. And so if you really want to cure cancer, what you really want to do is eliminate all cancer cells if possible. And what that means is if you need to be able to eliminate everything, you need to be also able to detect very small numbers of cells. It also predicts that if there are small numbers of residual cells that you can detect, that suggests maybe that the patient is at higher risk of relapse or might need a change in therapy or something like that. And so the question is, of course, also, if you can find very small amounts of cancer, does that mean patients need additional therapy? And that's still controversial, and that requires clinical trials, of course. So what are the various ways that we can detect residual disease? And I come from a hematopathology background, so I'm going to be showing you, in the heme path world, what we do. So one of the things we can do is look through a microscope. So this is a bone marrow biopsy, and it happens to be involved by lymphoma. You can see all these blue cells here next to the bone. So this is one technique, and maybe you can detect something down to, say, 5% involvement by lymphoma, for example, by looking through a microscope. So 5% is your detection rate there. We can enhance this detection rate by using immunohistochemistry. So we can take antibodies against molecules that are ideally specifically expressed on cancer cells and stain the cells with that. And so here's an example of a bone marrow which has been stained for cancer cells. You can see the cells here light up in red. So that increases your sensitivity in detecting things in a background that otherwise you can't really see them. Now, cytogenetics. Over here we have an example of a metaphase chromosome preparation. And so if you do this to look for cancer cells, typically in a cytogenetics lab, you'll look at 20 metaphases. So this is one metaphase. And so if you can find one abnormal metaphase, that's again 1 in 25%. It's not terribly sensitive. You can enhance your cytogenetic studies by doing something called fluorescence in situ hybridization, where you take fluorescently labeled probes to specific genes that are rearranged in the cancer you're looking for. And then you can look at more like 200 nuclei to see if they have a genetic rearrangement. So that gets you down to a sensitivity now of like 0.2%. Okay, so that's or actually 0.5%. So that's, you know, tenfold better. What else? Flow cytometry. Now, flow cytometry is something we can use in a hematopathology laboratory. And the advantage of flow cytometry now is you can look at very large numbers of cells. You can look at tens of thousands to up to maybe 1 million cells in an assay. And if you can use antibodies to detect your cancer cells, and we're talking primarily about leukemia and lymphoma in this case, but if you have antibodies that can distinguish your malignant cells from normal background, you can detect 1 in 10,000 abnormal cells. So this is quite a bit of an improvement in detection. Furthermore, if you do PCR, so over here we have a gel where we've done PCR, in this case looking for chromosomal translocation in follicular lymphoma. In this case, because no normal cells have a translocation, we can actually detect down to single cells with polymerase chain reaction, which in practical matters comes down to like 1 in 100,000 cells. So now we're getting very, very sensitive. Now, next generation sequencing, this is sort of the latest, greatest development in cancer. It turns out the next generation sequencing, as done sort of routinely, is not a particularly sensitive method. Whoops, I think I just messed up my Zoom. Hang on. And we're back, I think. Yes, okay, sorry about that. Yeah, next generation, on the other hand, you can design next generation sequencing assays to detect very specific sequence abnormalities. So we can do this in leukemia and lymphoma, looking for a specific sequence. And that actually can get extremely sensitive. So we're talking 1 in 1 million to even 1 in 10 million cells if you're looking for one particular DNA sequence. And then finally, I should also mention there is a lot of people are now working on looking for cell-free circulating DNA. So now we're talking about taking plasma from the blood and finding very small amounts of DNA in the plasma, and then using various molecular techniques to identify genetic abnormalities. And that possibly could be a minimal residual disease assay, although actually it turns out there's so little circulating DNA in most cases that it actually may not be really a minimal residual disease assay. So now let me show you just an example of how minimal residual disease can actually affect clinical outcome. So this is actually a figure that comes from a paper in 1993. So this is almost 30 years ago now. And this paper looked at using PCR to detect the 1418 translocation in follicular lymphoma, which is a particular chromosomal abnormality seen in this type of cancer. And what we're doing is we're taking patients who've had bone marrow transplants and looking to see what their outcome is, depending on whether the stem cells they got contained detectable cells by PCR. So what you see here is that in situations where the stem cells they got were negative by PCR, you basically have 100% survival. In contrast, if your stem cell products contain lymphoma cells determined by PCR, you have a really much more I mean, these are years, so it's not, these patients aren't dying rapidly, but the fact of the matter is that we don't have very good long-term survival. So this is really one of the first demonstrations of using minimal residual disease detection to predict clinical outcome in follicular lymphoma. Again, this is almost 30 years ago. This is a more recent study, actually from a laboratory here at the University of Washington, where flow cytometry was used to detect minimal residual disease in acute myeloid leukemia. And so again, it's almost intuitive that if you can detect small amounts of cancer, your outcome is going to be not as good. But in fact, to actually demonstrate that in a study is not simple. So in this case, they used flow cytometry to detect rare leukemia cells in bone marrow. And as you might expect, if you are positive by flow cytometry, your relapse is more likely to occur and your survival is not as good. And so again, it's demonstrating that minimal residual disease is really clinically important. This is an interesting case here. This is where they took children with acute lymphoblastic leukemia, and in this case, if they did not have minimal residual disease, they could detect no residual cancer, they actually treated those kids with less intense therapy. And what this basically is showing here is that the outcome of kids getting less intense therapy is actually no different than kids who got traditional, more intensive therapy. So what this means is not only can we use minimal residual disease to predict outcome, but we can also potentially tailor therapy to maybe have less severe therapy and therefore fewer complications, right? If patients are in fact have undetectable disease. So the examples I've shown you so far all have to do with hematologic malignancies. So what about with solid tumors? And the issue here really has to do with sensitivity. So as I already told you, flow cytometry, and again, we're looking primarily for hematologic malignancies here, we can detect one cancer cell in between 10,000 and 100,000 normal white cells. Now, in normal peripheral blood, you have about 10,000 white cells per microliter. And so if your minimal residual disease here is at this level, what we're talking about is between 1/10 and 1 cell per microliter, or about 100 to 1,000 cells per mil. And as I'm going to, and what's interesting is for solid tumors, we're talking about 2 orders of magnitude fewer circulating cells. So with metastatic cancer, we're talking about less than 1 cell per mil, or 1 cell per 10 million to 100 million normal white cells. So we're talking orders of magnitude difference in how frequently these cells may be found in the peripheral blood. And so what this means is you can't just use sort of ordinary flow cytometry. I mean, you could design a flow cytometry assay to detect circulating solid tumor cells, and they have molecules that blood cells don't have, as we'll see. But the fact of the matter is flow cytometry just cannot detect such small numbers of cells. So what it means is you have to do something to enrich your sample, your clinical sample, for tumor cells before you can hope to detect it. And so what can we do in that regard? So we've looked at this over the years, and many other people have as well. So here are some of the options for enriching peripheral blood for circulating tumor cells. So one thing you can do is get rid of all the white blood cells. So if you get rid of the white blood cells, and there are a number of ways to do this, that will give you a higher proportion of circulating tumor cells when you're all done. Another thing is by size selection. Solid tumor cells, when they're circulating, tend to be larger than blood cells, and so you can select for them on this basis. Immunomagnetic enrichment means taking magnetic particles, coating them with antibodies that specifically recognize circulating tumor cells, and then you can literally pull the cells out of the blood sample using a magnet. And this is the basis for the only FDA-approved circulating tumor cell assay that we have, and then we'll talk about that. Other people have done fancier stuff. You can use sort of nanofabrication to create devices that can filter circulating tumor cells, either based on immuno enrichment and or size selection. And what I'll talk about with the assay that we have validated, we're actually using density centrifugation. This is another way to sort of enrich for tumor cells and separate them from the vast majority of other cells in the blood. So this is just some data I will show you. Back in the early part of this millennium, we were doing some studies because we wanted to be able to detect circulating breast cancer cells using reverse transcriptase PCR. So what we were doing is we developed an assay to detect cytokeratin 19 RNA. And what we needed to do is enrich breast cancer cells in the peripheral blood to try to detect them using this PCR assay. So we tried two different ways to enrich circulating tumor cells. So one was using leukocyte depletion, and we used this stuff called Rosette-Sep, which basically is a cocktail of antibodies that stick to white blood cells, and then you can do density centrifugation to centrifuge away all the white cells, and then what you're hopefully left with are more circulating tumor cells. And so here you see blood that we spiked with different concentrations of cultured tumor cells. And this is actually flow cytometry, believe it or not. So in this case, we were just showing that if you stain for cytokeratin, which is the circulating tumor cells have, and CD45, which white blood cells have, we can actually get a pretty good enrichment of the circulating tumor cells using this leukocyte depletion method, and that's shown here in this box. We also tried using magnetic beads at the time, which predates any commercially available assays, but we used immunomagnetic beads coated with antibodies against epithelial cells, which circulating tumor cells are. And we also were able to see some enrichment of circulating tumor cells. But what we also saw, even here in the absence of circulating tumor cells, we found cells that appeared at least to be positive using this method. So we at least for this study opted to use leukocyte depletion because it seemed to be cleaner than using immunomagnetic separation. So what else can you do? So this is an example of how you might use size selection. There's something called microfiltration, where you basically pass cells through a filter, and if the holes are the right size, they'll trap the circulating tumor cells, which are big here in blue, and let the other red blood cells and white blood cells flow through. You can also make like a filter like this, where you have different slots on it, and in the slots themselves are the correct size to trap tumor cells. Or you can fabricate a device. This is just an example of a cutaway section, throwing a 3D device you'd make. And again, you've got this small opening down here. You put the blood in here, the circulating tumor cells being big get stuck, and the blood flows through. So these are just different strategies for enriching by size. Immunomagnetic enrichment, and this is the one that's used by the commercial assay, which is called CellSearch. In this case, you take this anti-EpCAM ferrofluid. So EpCAM is a protein that's expressed on epithelial cells, but not on white blood cells. And so if you have circulating epithelial cells, they'll be bound by these magnetic particles here, which are green. The white blood cells will not be bound by these magnetic particles. And then again, you can use a magnet to enrich for these circulating tumor cells. And ideally, you can get rid of most of these white blood cells. And so that's how that one works. And then these are some examples of microfluidic devices. And so what these all have in common is they select for circulating tumor cells partially by size, but also by, again, surface expression of this EpCAM molecule using antibodies. So this is one, this so-called CTC chip has these posts that they generated. The posts are actually coated with antibodies against EpCAM. And so the blood flows through, the circulating tumor cells get stuck. This is another example, just a different design of these 3D structures that allow things to pass through. And again, they also include immunomagnetic selection. And finally, here's kind of a cool one, which uses this flow through device. And this is a combination of magnetic selection as well as size selection. So in this case, you take magnetic beads coated with, in this case, antibodies against white cells, and the magnets cause as they pass through this device here, you have size separation based on these, like the path they have to go through. Plus you have a magnet that then pulls, in this case, pulls the white blood cells away. And so the circulating tumor cells come out of this hole and the white cells come out of that hole. So these are all very fancy, and people, obviously these are, you've got the references here for people who describe these things. So density centrifugation, this is the assay that we've actually been using. And I've actually been collaborating with Raresight now since the company's inception, quite a time ago at this point. And there have been a number of sort of generations of this technology that have been evolved, but the core of the technology is basically separating white blood cells and circulating tumor cells from the rest of the blood based on density centrifugation. And the way it actually works is you've got these special tubes that have a float, which has been manufactured to have a particular density, a particular buoyant density. And so when you spin blood in a tube like this in a centrifuge, what happens is the float basically separates white blood cells from red cells, neutrophils, and platelets, which float at the top. And what happens is then the buffy, what's we refer to in the hematology lab as the buffy coat, which are mainly lymphocytes, monocytes, and any circulating tumor cells, kind of end up between this float and the wall of the tube. So that's how this works. And we'll talk more about that in a moment. All right, so this is the only currently FDA cleared circulating tumor cell assay. It's called CellSearch. At this point, it has been passed down to about 3 different companies. We actually purchased this system in our laboratory in 2005. And so we had an instrument basically that we were using for over 15 years. And that's a long time to be using a laboratory instrument. And so when we got to the point where we were thinking about replacing it, it turned out that the technology was literally unchanged in those 15 years. So how does this technology actually work? So in this case, this is using immunomagnetic bead selection. So here you take blood and you mix it with these particles that have EpCAM antibodies on them. You then put this blood in a tube, and magnets on either side pull out the magnetically labeled tubes. You wash everything else away, and then you label the cells with fluorescent antibodies to distinguish epithelial cells from white blood cells, and inject them into this cartridge, which also has a magnet. And then it goes into this analyzer instrument here, and a fluorescence microscope makes images of the cells. And so what you're looking for here, in this case, we've got the EpCAM, one of the antibodies used stains cytokeratin, which identifies epithelial cells in green. And then the magenta that you see here is a DNA stain. And what we're looking for here for a circulating tumor cell is a cell that's green in its cytoplasm with a magenta nucleus. And so that's what a circulating tumor cell would look like. We might see some cells that have nuclei with no green staining, and those are basically white blood cells. And so that's how we distinguish circulating tumor cells from other cells in this kind of system here. This FDA clearance of this system is actually FDA cleared to detect circulating breast, prostate, and colorectal cancer cells. The data that was submitted to the FDA include looking at the number of circulating tumor cells and prognosis. So if for patients with breast and prostate cancer, if you have more than 5 cells in the 7.5 mils of blood that are drawn from the patient, then that portends a worse prognosis. Now, let me remind you, 5 cells per 7.5 mils of blood. Again, this is a very small number of cells. The clinical cutoff, it turned out, for colorectal cancer was only 3 cells per 7.5 mils. But yet these various techniques that can enrich for circulating tumor cells are actually able to find such tiny numbers of cells. So it turns out people have looked at it in the case, for example, of breast cancer or prostate cancer. The number of circulating tumor cells correlates with other tumor markers such as things like CEA and prostate-specific antigen. People showed that the number of circulating tumor cells correlates with radiological studies, particularly in the setting of breast cancer. And the response to therapy can be predicted by decreasing circulating tumor cells. If you treat a patient and the number of circulating tumor cells goes down, that suggests treating them correctly. And conversely, if you're treating a patient and the number of circulating tumor cells goes up, that suggests you might want to change the therapy. And so that's sort of the basis for why detecting circulating tumor cells could have clinical utility. All right, so here's just some data where people have looked at circulating tumor cells, in this case in breast cancer. So these 2 lines here, this is progression-free survival on the left and overall survival on the right. And so, for example, if you look at a patient with breast cancer, with metastatic breast cancer we're talking about here, pre-treatment, if they have fewer than 5 circulating tumor cells, they have better progression-free and overall survival than if they have more than 5 circulating tumor cells. So this is pre-treatment. If you then treat the patients and they come back and you test them again, same story. Fewer than 5 circulating tumor cells have better progression-free and overall survival. More than 5, less good survival. And finally, at the time of restaging, so after you treated a patient and you want to come back and see what they've, in this case, restaging traditionally would be done by radiological, imaging studies. Same story. More circulating tumor cells means poorer prognosis. This is not just true for breast cancer. So here it is for prostate cancer. Again, looking at survival in this case based on the number of circulating tumor cells. More tumor cells, poorer survival. This over here on the right is what we call a receiver operating curve, ROC curve. And it talks about how good the test is at predicting outcome. So if you had a perfect test, it would be 100% specific and 100% sensitive. And the ROC curve would basically go straight up to the top here, then across to the right. The green line here represents circulating tumor cells being able to predict outcome in prostate cancer. The red line is looking at prostate-specific antigen, which is sort of the traditional serum tumor marker for prostate cancer. And what you see here is that the area under the curve here, again, a perfect test would give you 100%, but the area under the curve here is quite a bit better for the circulating tumor cells than for prostate-specific antigen, suggesting that CTCs are a better marker than just PSA. And then finally, for colon cancer, which is the third type of cancer that people looked at with the rare site assay, not the rare site assay, the CellSearch assay, again, looking at progression-free and overall survival, in this case, the cutoff being 3 circulating tumor cells. Having more circulating tumor cells portends poorer progression-free and overall survival. So basically, these are the kind of data that enable the circulating tumor cell assay to get FDA approval, and is why, again, we think, although larger clinical trials are needed, we would hypothesize that doing circulating tumor cell assays actually has clinical utility in cancer. All right, so anyways, as I mentioned, we had been using the CellSearch assay in our laboratory, the FDA-approved assay, for 14 years. And again, when you get a 14-year-old instrument, it starts needing it starts breaking down a lot. So all of a sudden we had a lot more service calls. And sort of distressingly to us, because particularly those of us who work in academic medical centers, we always want to be sort of on the forefront of new technology. And the idea of replacing a 14-year-old instrument with essentially an identical instrument kind of rubbed us the wrong way in terms of we thought there should be some sort of new technology available. And the other sort of limitation of the FDA-approved assay is that it can only detect cells that express the cell surface molecule EpCAM, which is this epithelial cell adhesion molecule. And it turns out that in many types of cancer, the cancer cells actually lose their EpCAM on their surface as the cancer sort of progresses. There's something called epithelial mesenchymal transition. And that's a biological situation where tumor cells become sort of more, the best way to think of it, or one way to think of it, is that more stem cell-like. And they will actually lose their expression of the surface molecule that is necessary if you're using immunomagnetic selection to select for cells. And furthermore, if you wanted to develop a method for looking at circulating tumor cells that are not epithelial origin, we could talk about things like melanoma or renal cell cancer, for example. This is not useful at all, because if you don't have EpCAM on your surface, you can't use this assay. So, and finally, because it's FDA cleared, it turns out you can't really play with the assay. So what we like to do, again, in the academic world, is we like to see how we can tweak various lab testing systems to see if we can adapt them for new uses or improve them and develop custom assays, which is not an option with this. So for all these reasons, we thought about looking for other technology. And it turned out, actually, I'd been approached by the CEO of a company called Applied Precision in 2009. And Applied Precision was a company here in the Seattle area that specialized in high-end microscopy. And you'll see in a moment what this has to do with circulating tumor cells. And the question is, could we actually use this technology to detect circulating tumor cells? And the result is Applied Precision spun off another company called RareSite, and I've been working with them to try to validate some of this technology and see if we can make it work. And so this, again, I sort of talked about this already, but this is sort of the first generation assay that was developed. And again, you've got this float here with a particular buoyant density, and you spin blood in this tube, and the white cells, and again, any circulating tumor cells basically line up between this float, the very thin layer of fluid between the float and the wall of the tube. And so the initial idea of this assay was you take this tube then and just put it up next to a microscope. And this is where the high-end microscopy comes in. So you put it up next to a microscope, and if these cells are labeled with antibodies against epithelial cells and white blood cells and so on, kind of like the CellSearch assay, if you label them with similar antibodies, you can actually use fluorescence microscopy to image this very thin layer of cells between the float and the wall of the tube. And you end up with things like this. So here's an example of a tube of blood that's been centrifuged. Down here, you see the red blood cells at the bottom below the float. Up here is a thin layer of cells. These are actually platelets up here. And then in a band across the middle of the tube, this white band here actually consists of white blood cells. And again, this is where the circulating tumor cells will hang out, if any are present. And then if you basically take this tube and image this region and basically rotate the tube around, if you think of, you unwrap this layer around the tube, you end up with an image that looks like this. So this is basically, you can wrap this around the tube, and this is the buffy coat layer around the entire circumference of the tube. And then if you say, take this region, you look here, hard to see anything here, but if you look up close, now we can see, in this case, tumor cells stained with red. And this is how we can detect circulating tumor cells just on the surface of this float, just looking through the tube. Well, this is not the current technology. So this was okay. The images were reasonably good quality, but at this point, the technology has been modified to actually, after centrifuging the cells and getting the buffy coat layer, you can then put a band around the tube and essentially extrude the buffy coat layer into a little tube like this. And then these cells are actually smeared onto 8 individual glass slides. These are ordinary microscope slides like any pathology lab would use. Then you take these slides and put them in an automated stainer. Again, these things are routinely available in pathology labs, and stain the cells with the various fluorescent antibodies we've talked about. And then this is the instrument that actually images the slides. You get these very beautiful pictures that look like this. So in this panel, you see the cytokeratin EpCAM stain. So this is identifying epithelial cells. In this panel over here, the blue stain you see are just nuclei. And then finally, we also have the stain for CD45, which identifies white blood cells. And if you put all these images together, you end up with something that looks like this. And there you see right in the middle, one little lonely circulating tumor cell surrounded by a sea of white blood cells. And we get eight glass slides out of this assay. And so we can actually detect single-digit number of cells by scanning all these slides with immunofluorescent microscopy. And so here's just some examples. So here again, we have breast, lung, and prostate cancer. And here you see the cytokeratin EpCAM stain. So these are the epithelial cells. These green cells are the white cells, and then blue are the nuclei. And the cancer cells are the ones that stain for cytokeratin and EpCAM and have a nucleus and do not stain green. And so we can find these cells. Now here's a picture of actually a cluster of tumor cells here. So in this case, the cytokeratin cells are green, EpCAM is red, and the CD45 is in orange. So there we've got some white blood cells here. This is actually a cluster of tumor cells circulating in patients with cancer. And this is actually something we see not uncommonly in patients with metastatic cancer of various types. Cells actually circulating as clusters of little bits of tumors that circulate in the peripheral blood. So we next wanted to actually validate this test for clinical use. And so validation has a very specific meaning to us in the clinical laboratory. And this is the process you have to go through if you're going to offer something as a clinical test for patients. So one is sensitivity. Sensitivity means if a disease is present, what percent of the time can you detect that? You want it as close to 100% as possible. Specificity. This refers to do you ever get a positive result in a patient without the disease? You don't want that to happen. So you want to be basically 100% specific. The next thing you want to know about is precision. So that is if you do an assay on a sample repeatedly, how close are the results to each other? You want this to be very close to each other, have very little variation from one assay to another. Another is recovery. So what this means basically, if you take a sample and you have, let's say, circulating tumor cells, for example, that you want to detect, if you know that the sample has 100 cells in it, what proportion of the cells do you get back after doing your assay? You hopefully get all 100 cells back. Linearity refers to, as you look at various numbers of tumor cells from 0 to a high number, do they fall on a line? So that's important. Interferences in the laboratory, we always worry about whether patients have things in their blood that could interfere. In the context of circulating tumor cells, we don't worry about this too much. On the other hand, if you're looking for basically certain lab tests require color reactions to occur. And then when the color reactions occur, that can be interfered with things like bilirubin or hemoglobin. So but this is less of an issue for us. And then finally, accuracy. And the way you judge accuracy is you do your test and you get a result and you compare it to another test that you believe also gives you a correct result and see if those results agree. So I'm going to take you through this process and show you what we've done in terms of validation. So the first thing we looked at is sensitivity and specificity. And so what we did is took cultured breast cancer cells, they're called SKBR3 cells, and in 5 tubes of normal blood each, we either put 25 cells, 50 cells, or 125 cells. We looked at all the samples, and in no case did we miss tumor cells. There are always tumor cells detected, which basically give us 100% sensitivity as sort of a yes or no kind of answer. Importantly, however, this is actually more important, in the normal blood samples that were not spiked with tumor cells, we did not find any signals that made it look like there were circulating tumor cells. And so that's really specificity. If you're looking for cancer, you really don't want to have false positive results. So that's very good. Now, it turns out from a technical standpoint, it's actually very difficult to spike normal blood with very tiny numbers, like single-digit numbers of cells, like you might find in patients. And so to look at patients who look for lower cell numbers, we basically just had to use clinical samples. And so we were lucky to have patients in our clinic who agreed to participate in our study, and so we were able to do this. In terms of precision, we're taking, again, these same 5 samples spiked with what we think are the same numbers of cells. And as in many laboratory assays, if you have a small number of things you're looking for, you tend to have a fair amount of variation in the number that you get out. But by the time we get up to a higher number of cells, we have relatively good reproducibility. So again, when you're looking at rare events, you tend to have a fair amount of variability. We'll talk about that also, because that's actually, you come up against an actual mathematical limit to what you can do. And we'll talk about that. Here's the linearity studies. So again, these are our replicate samples of spike-ins. And if you draw a line through the mean of these 5 replicates, you get a line that gives you a slope of 0.994, which is very close to 1, which is what you want. Your intercept is a small number, and your R-squared, your correlation coefficient, is 0.96. So this is what we would consider to be quite good reproducibility assays. In terms of recovery, so this is where we put, can't talk now. This is where we take normal blood samples and we spike with a known number of cells and ask how many of the known number of cells do we actually get back in the end. And again, for all these different spike in numbers, we actually got very close to 100% recovery. So that's quite good. As far as interferences, I mentioned that it's probably not a big issue. We looked at bilirubin just because it's traditional, and we found no interference with bilirubin. And triglycerides is another thing that traditionally interferes with laboratory tests. No interference there. I mean, if you think about it, because there's this washing and cell preparation, things like this, any types of small molecules like this should be washed away by the time you get to making your slides, so they shouldn't really interfere. So this is no surprise. How about accuracy? So this is really the sort of the thing that we really care about. Our gold standard in this case is the FDA approved CellSearch assay, and we did a comparison here. We had 48 patient samples to look at, and so here are the numbers that we got using the so-called predicate method, the traditional assay. Here is our assay that we were trying to validate, and the numbers here actually look quite good. So we have a slope of 1.3. We have an R squared of 0.9 and so on. So that looks great. But if you look at it closely, you'll notice that most of the data are clustered right down here near the origin. And so doing just linear regression like this on a linear scale is really probably not something to do with this kind of data. So we did a log transformation. And what a log transformation does for us basically is spread out the lower numbers so we get a more linear, or at least we get a better distribution of the data, shall we say. One of the things that I also have to tell you about here is, if you go back and look here, go back here. There's a lot of cells with 0, because again, these are patients with circulating tumor cells, patients with cancer, and they don't always have circulating tumor cells. You can't log transform 0. So what we did was, this is a common thing that you do when you have data like this, is we basically add 1 to the numbers from each sample. And so that allows us to do a log transformation. And now we get a curve that looks like this. And that you'll say, well, this is not quite as pretty as the previous curve. But the fact of the matter is, we actually get a reasonably good slope and a reasonably good R squared given the scatter down here at the low end. And so at least we accept that, from a laboratory standpoint, this is reasonable correlation. And at the higher end, we get very good correlation. And we'll address the low numbers here, because this degree of variability is inherent to the fact that we're looking at small numbers of events. We'll get to that in a second. Now, how about accuracy? So what we're looking for, these are a number of tests to look for whether you get your data agree with each other. And these tests tend to be used to look for whether data are different. And so the fact that we have p-values that are not clinically significant, or that are not significantly different, suggests that the two data sets that we have, and these are all pairwise comparisons, are essentially the same. And then finally, this is accuracy in terms of looking at the prognostic cutoffs. So if you remember, for breast cancer and prostate cancer, if you have more than five or more cells per tube of blood, that puts you in a poor prognostic category. And if you have less than 5, that puts you in a better prognostic category. So we just asked, of our 48 samples, how many agree as to whether you are positive, i.e. poor prognosis, and negative, i.e. good prognosis. And the good news is we actually get pretty good accuracy. We get something like over 90% concurrence between the two assays. The positives by one assay are the same, or agree with both. The negatives are also. But notice there are 6 samples here in which there's disagreement between the two assays. So anyways, from a statistical standpoint, this is actually great. We got a great p-value. This is great from a chi-squared analysis. But the question is, how much do we need to worry about these assays that are disagreeing? So this begs the question, how reproducible can a CTC count be? And we get into the realm now of Poisson Statistics, okay? So now let's say that a patient's real life CTC count is 5, and they're absolutely, they're at the cutoff of 5 in real life. The fact of the matter is, if you draw a tube of blood from this patient, say 100 times or 1,000 times or more, you'll get a distribution of numbers that look like this. This is the Poisson distribution. So, most of the time, or the median at least, will be 5, but there's a very wide spread here, and this is strictly due to sampling, not sampling bias. This is just due to the random nature of, you stick a needle into a patient's tube, and they actually have 5 cells per tube of blood. you're going to get a distribution like this in real life. This is the Poisson distribution. This gets even worse with 3 cells. So anyways, if the patient's true count is 5, 95% of the time, it's going to be somewhere between 2 and 9. So this is a pretty broad number here. And notice that this number overlaps with the prognostically significant number. Same thing if your CTC count is 3, if you've got colorectal cancer, 95% of the time, you're going to find between 1 and 6 cells. So half of the time, in both these cases, you're going to make a patient look like they're not bad prognosis, when in real life, maybe they are. So this is literally a mathematical limitation of any of these assays. It doesn't matter how good you are. It's really just a matter of randomly sticking a needle into somebody's arm, pulling out 7.5 mils of blood and saying, did I get a cell or not? Okay, so now bearing that in mind, these are our quote unquote discordant cases. So these are the 3 where it was more than 5 cells by CellSearch and less than 5 cells by our assay. But if you look at, if the true number is the mean, let's say, of these 2 numbers, here's your 95% confidence intervals. And you'll see here is that if you take this range as reality, these methods actually agree. And the same thing is true in the reverse direction. If we found more than 5 or more cells by Rarityc and less than 5 by CellSearch, again, based on the sort of mean number here, the confidence intervals actually indicate that these are actually could be the same number. Which I guess from a philosophical standpoint is if you're going to be making clinical treatment decisions based on a cutoff of 5 cells, you're going to have a significant amount of time where you might be getting the wrong answer. Finally, this is another way of looking at accuracy. This is a bland Altman plot. And so what this plot does is you compare the 2 results of your 2 different methods, you subtract them from each other. If they're the same, of course, the subtraction gives you 0. And if they deviate, that means the test isn't so good. So what you see here is at the low end, we actually have very good agreement between our 2 methods. They start to skew out a little bit here, but these dotted lines basically give you 95% confidence intervals that basically the things agree. And so we have very good agreement here, but look at this over here. What is that? We've got this one sample over here, which has something like over 200 cell difference between the 2 methods, in which case we found something like 500 cells using the Rarityc method, but on the order of 200 or 300 cells using CellSearch. So what's going on there? Oh yeah, here are the actual numbers. 548 Rarityc, 340 CellSearch. That's a pretty big difference. Well, it turns out that in this particular patient, only 25% of their circulating tumor cells had detectable EpCAM. And if you recall, the CellSearch assay requires EpCAM expression in order to find the circulating tumor cells. And so we think, and we'll just show it right here, this is the EpCAM signal here. So this patient's cells stain quite brightly with cytokeratin, which is shown in this column, but you can see they're pretty weak for EpCAM staining. So that is the most likely explanation as to why there's a discrepancy between the 2 methods. Okay, so I'm going to take the last few minutes just to talk about some other issues here that aren't relating to our assay. But the question, for example, what's the difference between circulating tumor cells and cell-free circulating DNA? Cell-free circulating DNA is a lot of people are investigating its use for, particularly looking for mutations in tumors without actually having to biopsy tumors in a patient. These are the so-called liquid biopsies. Both of these really are liquid biopsy techniques. And so the question is cell-free DNA telling us something different from CTCs? Are they basically equivalent? And so in this paper, what they did was they looked basically at a whole bunch of different gene mutations by next generation sequencing and looked to see what's the correlation between mutations found in circulating tumor DNA versus circulating tumor cells. And what this basically shows you is there are a lot of genes that are, so these dots here are basically where the 2 agree. But you can see is there's a lot of differences also in the genes that are detected as mutated in the circulating tumor cells as opposed to the circulating tumor DNA. And what this would suggest is there's probably something biologically different between circulating tumor cells and just all the cancer that may be present in the patient. And I would just argue that, there's sort of a disagreement. Well, why do we need to look at circulating tumor cells? We can just look at circulating tumor DNA, but there actually are differences between the two. So they probably are complementary techniques. So what else can we do? So far, what I've basically been talked about are counting cells. How many circulating tumor cells are they? But we can actually do some additional studies to look at some of the biological characteristics of these circulating tumor cells, as well as just their number. So for example, you can look at prostate cancer to see whether they express androgen receptors or even particular variants of androgen receptors. You can take breast cancer and look for estrogen or progesterone receptors or HER2 in breast cancer by staining for those. One thing you can do with this technology is actually study single cells. So there are ways of picking single cells off of these glass slides that I talked about and doing single cell analysis on those. We're also, there are also, for example, in the research world, clinical trials, we've been working with some investigators to look at certain markers. For example, there's this marker called MUC1 star in triple negative breast cancer that is a potential target for CAR T cells. So basically designing immunotherapy to detect these cells. The other thing we could potentially do, and we haven't started work on this yet, is looking at non-epithelial malignancies, because again, we don't have to worry about EpCAM expression. So here's some examples of additional biomarkers you can look at. So here's an example of a breast cancer cell. Here's the cytokeratin EpCAM, and here you can see it expresses estrogen receptor as well as HER2. In prostate cancer, again, you hear the circulating tumor cells with cytokeratin. This is their androgen receptor, stained here in yellow. And this is a splice variant of androgen receptor that has some clinical prognostic importance, ARV7. You can stain for that as well. Furthermore, you can look for checkpoint markers. So these are markers that are used for immunotherapy. And so again, here's your cell. In this case, we're looking at PD-L1. In this case, RF1, which is also known as MUM1. This is just another lineage marker, showing that you're not just limited to looking at cytokeratin and EpCAM. You can look at other biomarkers that might affect therapy or prognosis as well. One of the things that we are looking at more closely is prostate cancer, partly because this is a research interest of a lot of our colleagues over at the Seattle Cancer Care Alliance. So one of the things, this variant, the splice variant of androgen receptors is one thing that some folks are looking at. It can be looked at by immunofluorescence staining. You can also look at it by RT-PCR. But one of the other things that we are looking at is prostate cancer in which there is what's so-called neuroendocrine differentiation. And it's thought that these prostate cancer cases that undergo neuroendocrine differentiation may have a different biological behavior, need different therapies, for example. And the marker that we use for that is called synaptophysin. And so this is one of the things that we're actually working with now with one of our colleagues at the Seattle Cancer Care Alliance. So this is just showing some staining for synaptophysin, as well as this ARV7 version of the androgen receptor. Again, these are research studies that we're working on, but the difference here actually between what we've been doing so far with enumerating cells, looking at some of these markers, is we can start looking at things like the actual level of immunofluorescence staining, the immunofluorescence intensity of some of these cells. And so we've taken various cell lines that express various levels of these markers. So here again, a synaptophysin, here's a low expressor, here's a high expressor, and so on. Here's androgen receptor, high expressor, low expressor. And so we've looked at this now a little bit more quantitatively. So here's an example of looking at the quantitative expression of synaptophysin, comparing a high expressing cell line versus a low expressing cell line. So again, we're not just counting cells anymore, we're actually looking at the levels of the proteins in these cells. And here are 3 other cell lines for comparison. We can do the same thing with androgen receptor. Again, high expressor, low expressor, and here are 3 different cell lines that have various levels of expression. And we're hoping that, and then we just look at sort of sensitivity and specificity for these. Here is some precision data. unfortunately, I don't have time to go through this in the same detail as we did our validation, but basically what we're doing here is showing you all the validation data now for some of these alternative markers, but not just looking for positive or negative, but actually looking at levels. I'll also show you one slide here. Again, it is possible to pull single cells off of these glass slides, and then you can subject these single cells to things like whole exome sequencing, or you can do array CGH on single cells picked off the slides. So this is something, again, in the research world can be used. It's not really a clinical assay, certainly at this point. And so just to tell you what some of the things that we're looking at, I've already talked about this MUC1 star. So again, we're looking at hoping for CAR T cell therapy in this case in breast cancer. And we're trying to we're developing collaborations again with some of the clinicians treating melanoma or renal cell cancer to see if we can look for CTCs in those cases. So finally, I would like to thank the people who I have worked with over the years. In our department, Mike Tam, Michael Harden, and Sarah Trimble are all folks from our clinical laboratory who actually did the validation work with their own hands. Elisa Klein has been an invaluable member of our team. She's our clinical coordinator who has recruited patients without whom none of these studies would be possible. And Jeff Baird is our department chair. At the Seattle Cancer Care Alliance, we've worked with Tia Hagana, who is a prostate cancer doc, BK Gotti and Evan Yu are breast cancer docs. They, again, have helped us with their patients who, again, we can't do these studies without. And finally, these are the folks at RareSite who have been our collaborators over the course of this collaboration. And Ron Subert in particular was the founder and CEO, who's now retired, but none of this would have happened without him. So at that point, I'll stop. We're just almost at time, but I'd be happy to answer any questions if anybody has them.
Thank you. We do have a couple questions that came in, and if you have not submitted a question, you can do so via the Q&A box, which you can find at the bottom of your screen. So first question for you, Dr. Sabath. It's two parts. How much greater sensitivity improvement is obtained by utilizing a pan-CK antibody, and are there any CKs not cross-reactive with that antibody?
Let's see. So to answer the first, I mean, I'm not sure we know the answer to either of these questions in particular because we never really did a comparison between various cytokeratin antibodies. I mean, in the clinical realm, if you're looking in a piece of tissue for epithelial cells, the pan-cytokeratin antibody is really it really reacts with most epithelial tissues. There are some exceptions, and so the epithelial antibody used in this case sort of corresponds to one of those. It's not one of these sort of specialized specific cytokeratin kinds of things. I would imagine there probably are some cytokeratins that are not cross-reactive, but those would be rare cases. But I don't have any actually hard numbers for that, so I apologize.
That's great. And next question is, what is the percentage loss of cells with the cytospin?
So that gets at the recovery data that I showed. And so it's really pretty low. I mean, we have really close to 100% recovery, at least when you do sort of artificial spike-in type experiments. And so I guess the answer is we really have pretty good recovery. And so we have very little loss in the sort of the sample preparation stage.
Wonderful. And are the cells used live cells?
Are the cells used? I mean, they're not by the time you get to the imaging them, right? Because you have to, I mean, they're initially live when you do the centrifugation, and they haven't been fixed or anything until you actually apply them onto the glass slides. So prior to application onto the glass slides, they have not been fixed. Oh, wait, I take that back. The collection tubes that we use actually do contain a preservative, so the cells are not alive. My bad. It's theoretically possible to use live cells with this kind of methodology, but that's not how the clinical assay works. Yeah, so the special collection tubes that we use contain a preservative, which is basically sort of a mild fixative. What that means is the specimens themselves are actually quite stable. We can analyze them 3 or 4 days out from collection, which is great from a clinical laboratory standpoint. If you're interested in recovering viable cells, however, you would have to collect them into some other kind of medium like EDTA, for example. You could probably do the cell preparation step because that, I mean, it hasn't been validated, and so I don't know how it would work, but it should be theoretically possible to at least keep the cells alive for part of the process, but that's not something that's really been a major focus of what we're working on.
Wonderful. And I'm going to squeeze in one more question here. And you did touch on this a little bit. How many cells do you typically find in a cancer patient?
That's a great question. Of course, that's sort of $1 million question from a clinical standpoint. So in patients with metastatic disease, I mean, it's more likely you're going to see it in patients with metastatic disease, and typically those numbers are very low, like single-digit numbers of cells. When patients start developing very high numbers of cells, it's extremely, bad from a prognostic standpoint. And we have, unfortunately, a lot of our patients end up passing away who have, very large numbers of cells. In patients with early stage disease, and we're talking stage 1, 2, and probably 3, I would say greater than 99% of the time you're going to find nothing. So this really is an assay for dealing with patients with metastatic disease, who again are the ones who have the most dismal prognosis, and the ones we need to work hardest on trying to find better therapies for.
Wonderful, thank you.